Assalamu alaikum. So we will be discussing somatic embryogenesis in plant. How embryos are formed in somatic cells. How embryos are formed in somatic cells. So we can say it's the formation of embryo from the zygote. Embryogenesis is a process, but the initiation and development of embryo are formed from the zygote. This embryogenesis is of two types. Embryogenesis is of two types. Zygotic embryogenesis and somatic embryogenesis. Zygotic embryogenesis and somatic embryogenesis. Now, what is a zygotic embryogenesis? It is the formation of embryo when there is a formation of zygote. When two male and female gametes come together, they fuse together and form a zygote. Male and female gametes are in N form. When they fuse together, they form a 2N cell. So this is the zygotic embryogenesis. In case of somatic embryogenesis, there is no fusion of two gametes. We take somatic cells which are going to form the embryo. Stewart was the one who discovered this formation of embryogenesis in case of carrot. Stewart in 1958 was the one who discovered the formation of somatic embryogenesis in Dicus carota. When the explant forms the small lumps, these are, we can call it, when they go for the formation of the embryogenesis, that process, these lumps are called as embryoids. When the explant is undergoing through the embryogenesis formation, they form the small lumps on the callus. These small lumps are actually the embryoids. These are actually, or the embryo, we can call them embryo. But in case of plants, we call embryo as embryoids. But these embryos are formed when there is availability of proper nutrients and the environmental conditions. Here we have discussed the both organogenesis and the somatic embryogenesis. We are taking the plant as here from anywhere, either leaf or stem, we can take our explant. This explant can cultured on our medium, then they form the callus. Then on the above of the callus, they form a small structure. These are called as somatic embryos. Explant से directly small embryos बनाते हैं. We call this process as a somatic embryogenesis or direct somatic embryogenesis. When the explant first form the callus, then from the callus they form a small lump-like structure on the callus. These are called as embryoids. And if we will take this individual look, this is our somatic embryoid via the indirect process. From these somatic embryoids can be used for regeneration of the plants. This is the whole for the embryogenesis. Even you can see here organogenesis also. Like we have taken the explant, we can go for, from the explant we can directly form our root or shoot, depends on your culture medium. This type of organ formation is called as direct organogenesis, or we can form intermediate step first, that is the from explant to callus, then callus to regeneration of root or shoot, depends on the medium which you have taken. For rooting, you have to take more oxygen than the cytokinin, and for shoot process, you have to take cytokinin more to the oxygen process. This is the whole regeneration pathway, how plants are able to go for organogenesis or embryogenesis. Even it's like somatic embryogenesis is not, every explant can't be used for this formation of the somatic embryogenesis. Scientists have given some list where we are able to form our somatic embryos. Like we can use immature zygotic cells, where we can use mature zygotic cells. Inflorescence of flowers we can use, or we can use cells which are in cell suspension culture or in single cell. Protoplast we can use, root, stem, or leaf we can use as a explant for the formation of the somatic embryos. We know that growth hormones play an important role, as we have seen in organogenesis, that if we have to go for the formation of the root, then oxygen concentration should be high or as compared to the cytokine. Or if we have to go for the formation of the shoot, that time we have to take cytokine more than the oxygen. But here we are having something different, some different approaches. Like oxygen, when we are placing our explant onto the medium, it should be only having oxygen first. 2,3-D is our commonly used oxygen, synthetic oxygen it is. So what it induces? It induces the cell elongation and cell division. It induces the cell elongation and cell division. It induces our cell elongation and cell division. That means when cell is going to divide, it induces embryogenesis. But when it induces its cell division elongation, when it started to divide, when certain cells are formed, that time then again we have to take these cells into the medium which is devoid of oxygen. क्योंकि जो ऑक्सीजन है वो फिर क्या बनाएगा, that is going to form the callus. That is not our embryos. Or what we have to do when the cell division has started, we have to take this sample, then we have to place it on another medium which is devoid of oxygens, and then only embryos will be formed. Or if the oxygens will be continuously there, then no embryos will be formed. In certain, like we all know that different explants are having different nutritional requirements. It varies. So in some cases, what scientists have done, they have used gibberellic acid to induce the embryo formation or embryoid formation. But in certain cases, it has seen that they are having reverse effect, that if gibberellic acid is present, then no embryo formation will take place. This was about the role of the oxygen in embryogenesis. Now, the second example is our cytokines. The most commonly used cytokine in embryogenesis or zeatin. When there is the formation of embryo, because we know that we have studied this thing, structure of embryo, how the single cell is getting converted into the complete embryo, or how seeds are getting formed. So this cytokinin is responsible for the globular embryo. Above of the slides, we will be able to see what are the different stages of our embryo. So this zeatin is responsible for the formation of globular embryo. Now, even it has seen that ABA, abscisic acetic acid, also promotes embryo maturation. When there is the embryos are formed, that time we have to treat our embryos with the abscisic acetic acid. It means they will go for the embryo maturation. Now, what are the different factors which are going to affect our somatic embryogenesis? It is recommended that we have to use reduced form of nitrogen for embryo to induce the embryo formation. So whenever we have to take the nitrogen source, we have to take nitrogen source in the reduced form, that is in the form of nitrate. Then in the form of nitrate. Then another important parameter is there, that we have to add polyamides. We have to add polyamides for the development of somatic embryos. Now, what are polyamides? They are also having their own class. They come under the class of growth regulators. These polyamides are actually the organic compounds. They are having organic compounds, plus they are having more than 2 amino groups with low molecular weight, And the best example is their alkyl polyamides. These polyamides occur freely in the plant, and we are having certain examples of polyamides like putrescine, spermidine, and spermine. Putrescine, spermidine, and spermine. Putrescine have shown that it increases the formation of embryogenesis. Even in certain plants, it has shown that while giving some electric stimuli, they promote embryogenesis, and this type of effect of stimuli has been observed in alfalfa and tobacco plant. When we have taken alfalfa and tobacco, we have exposed these plants to the mild electric current, that is 0.02 volt DC for 20 hours. Then it has been seen that there is the formation of embryoids. The electric stimuli promotes the differentiation of organized shoot or embryo by affecting their cell polarity through the change in the organization of microtubules and induction of asymmetric cell division. So we can say that it induces asymmetric cell division, then the embryogenesis is promoted. We are having now the stages of embryogenesis. From the explant, we have to form the embryoids. So first is the induction, second is the development, and third is the maturation. And for somatic induction or somatic embryogenesis induction, we have to introduce auxin 240. It's a synthetic auxin into the medium. The auxin should be relatively higher concentration so that when abruptly it starts the cell division. Then when it has started the cell division, then cell is continuously growing. Then the second is the development. In developmental process, the medium should be auxin free. That time this explant has developed into the mass of cells. This mass of cells is called as pro-embryonic mass or pro-embryonic cells, PEM. Pro-embryonic mass. जब ये इंड्यूस हो गया सेल डिवीजन, फिर इसके बाद हमने इसको डेवलपमेंट के लिए कहाँ रखा? ऑक्सीजन फ्री मीडिया में, जहां पे ये कंटिन्यूअसली मास ऑफ सेल्स बना रहा है, उनको हम लोग क्या बोल रहे हैं? प्रो एम्ब्रियोनिक मास ऑफ cells. Then the third process is the maturation phase. This maturation phase for somatic embryogenesis is very poor. This plant has not shown good result for at maturation stage. Embryos should be able to, what is maturation phase? When we have formed the embryo, it should be able to reserve their food material and proteins. It has been seen that abscisic acid promotes normal development of embryogenesis. When the third process has to be carried out, it should be carried out in the presence of ABA, growth regulator, as it develops the normal development of the embryogenesis. Now here I have taken the example of dicot. This embryo formation. So different stages are there. You can see how the embryo is formed. First is the egg, which is a single cell. Then from the egg, we are having another stage, that is our 2-cell stage. Then the octet stage means 8 cells bani hai. Then from the 8 cells, there is a formation of glober cells. Then from the glober cells, we are having triangular cells, heart-shaped, then torpedo cells, then lastly the cotyledon stage, or simply the formation of the seed. But in case when we have placed our explant under in vitro condition, or when we are using the somatic embryo, that time from the callus, From the callus, the globular stage is formed, this stage. Then from the globular stage, heart shape is formed. Then the heart-shaped torpedo stage is formed. Then from the torpedo stage, a plant-like structure is formed, which is going to convert into root and the shoot. Again, I will repeat. This is the for somatic embryogenesis. First the callus is formed. From the callus, globular structure is formed. Then from the globular structure, heart-shaped cells are formed. Then the torpedo, and finally the structure which is going to form the root and the shoot in case of somatic embryogenesis. Now, the types of the somatic embryogenesis. Somatic embryogenesis can be carried out via the direct method and the indirect method. In case of direct method, what we are doing, we are taking our explant, onto the solid media, So the medium should be having the MS media should be there. It should be agar should be there because it's a solid, and we have to add oxygen, that is our 2,4-D. Then it means the induction of somatic embryos takes place. Then we have to place, or these cells which are growing here has to place in another media for the development process where there should be oxygen free. Then the globular elongation and maturation stage of somatic embryos takes place. This is how the direct method of somatic embryogenesis is carried out. Now, the another example is there, somatic indirect method for somatic embryogenesis. In case of indirect method, we have to go for the formation of the callus. So MS media 2,4-D is there. When the callus is formed, then we have to take this callus into a liquid medium. We have to take this from this callus. We can go the somatic embryos. Then different stages of cells are formed. This is our the indirect embryogenesis. Means हमें इसमें बीच में क्या बनना चाहिए, callus बनना चाहिए, तब जाके हमारे जो elongated या globular stages है वो बनने चाहिए. So even this somatic embryogenesis जो है, जो हमारे somatic embryos है, we can use them as a seeds also. These are called as synthetic seeds. जो छोटे छोटे embryoids बनते हैं हमारे callus में, we can use those as a synthetic seeds. These synthetic seeds has to mix with the calcium alginate matrix. So here sodium alginate and somatic embryos are formed, mixed together. Then this mixture, sodium alginate plus somatic embryos, are dipped into the calcium chloride solution. Then they are dipped into the calcium chloride solution. Then again from here calcium chloride, they have to place for dehydration process. We have to place in such a solution where dehydration takes place, that is our sucrose medium. So our synthetic seeds are placed in the sucrose solution for dehydration. Then we have to take our dehydrated artificial seeds because they are coated with the alginate. It's a gelling agent. तो इनके ऊपर क्या होता है, gel like structure होता है इन embryoids के ऊपर. We can store them at 4 degrees centigrade. Then again, when we are in need of these seeds, we can use these synthetic seeds or artificial seeds. We can grow them on MS media with the effect of gibberellic acid, gibberellic acetic acid, gibberellins. We can use GA3 because GA3 induces, or this gibberellin induces the formation of radicle and plumule. When the radicle and plumule are formed in vitro condition, then we have to take this explant onto the soil, and this process is called as the hardening. When these plants adjust in this soil, then from here greenhouse effect, then to the environment, natural environmental condition, we can place over this plant. Now the advantages of our somatic embryogenesis. What are the different advantages of carrying out this somatic embryogenesis? We don't have, we can manipulate our experiments as per our need. These in somatic embryogenesis are having high propagation rate. They grow fast under in vitro condition. Even it has been seen that there is a lot of variation at the genomic level, that is our somochloro variants are formed. Even germplasm conservation, we can store these synthetic seeds for germplasm conservation. Even they are labor saving. When we are having an infected plant, that time we can take a cell from this infected plant which is not having infection. Then from that single cell, we will be able to go for micropropagation. Everything has its own disadvantages, as the somatic embryogenesis is confined or fixed to fifth species. Only fifth species has shown, or they are having positive approach for the formation of the somatic embryogenesis, because the somatic embryos show very poor germination rate because of their physiological and biochemical immaturity, because they are not the complete zygote. So there are certain physiological and biochemical maturity. Even it has been seen when we store these synthetic seeds for longer time, they show the instability. So we can't apply this technique at a commercial level. We can't apply this technique at a commercial level. So we are having its own limitations also.